Hello and welcome to Unit 2. Unit 2 is talking about biochemistry and cell biology. And this first video is going to give us some introduction onto some of the chemistry that we need to understand in order to understand the bigger picture. Okay, so if you have a quick look at this list, if you've done chemistry courses already, then most of this stuff will be familiar with you, but we'll go through some of the definitions of this stuff pretty quick because these are the terms that you'll need to use to understand the chemistry. I'm not going to spend a whole lot of time on intro chemistry because you're going to get more detail on this when you actually do some chemistry courses. But let's go through it so that you get an understanding of what we're talking about. Okay, when we're talking about chemistry, basic chemistry or biochemistry, we start off with atoms. Okay, atoms are the smallest units of matter that retain the characteristic of an element. Okay, in chemistry is the study of matter. So they start off studying atoms. Biology is the study of life, so we start off studying cells. In order for us to understand what a cell does, we need to understand its components, what makes it up. You remember, a cell is orders of magnitude bigger than an atom. So if we understand the basic chemistry, then we've got a better idea about how that cell works. So if we look at an atom, we can see that an atom is composed of a nucleus, and inside that nucleus it has things that are called neutrons, which are neutral or have no charge whatsoever, and we have protons, which are particles that have a positive charge. On the outer shell of the atom, we're going to have electrons, which have a negative charge. Now, in a normal stable atom, you'll have the electric charge on the outside, and the electrons will be the same as the protons in the nucleus. So the nucleus, of course, is at the atom's center, and it has protons, which are positively charged, and neutrons, which have no charge. So we just said that a second ago. Now, the electrons are what kind of make these atoms interesting. They're negatively charged. They orbit in energy levels outside the nucleus, and then they determine an atom's chemical reactivity. So the chemical reactivity is what's kind of important for certainly chemists, but definitely biologists when we're trying to understand how these things all work and come together. Now, within the atom, a number of protons equals the number of electrons, and that gives us the atomic number. So if you look at the periodic table, you'll see every atom, every element up there has an atomic number, and that's generally equal to the number of electrons and neutrons together. So if we look at carbon, for example, which we are a carbon-based life form, so carbon is super important to our structure. Carbon has the atomic number of 6. So that should tell you that carbon has 6 electrons on the outside and 6 protons in the nucleus. Now, an atom is composed of various energy levels which kind of orbit around the nucleus. And these energy levels are called valence shells, and these valence shells fill with electrons. So this is the general basic structure of an atom. Like I said, you're going to get a lot more of this when you do your chemistry courses. Now, radioactivity and isotopes, this is stuff that's going to be really important to your course, and you will definitely get a lot more of this as you move through courses in your program. So if we look at the atom, keeping in mind the neutrons and the protons within the nucleus, if the number of neutrons and the protons are different, then we have an isotope. So an isotope is an element that has the same atomic number as another form, but the atomic weight changes because there's differences in the neutrons. I hope that makes a little bit of sense. Some isotopes can be quite stable. How other isotopes, forms of the same element but different atomic masses, they can break apart at different times and they can give off atomic particles. These isotopes are said to be radioactive. Okay. Now, radioactive particles do exist naturally. We do find them. If you go to places, you can measure radioactivity that the Earth naturally emits. But many of them are man-made. So we have made more in the lab than we find that occur naturally. Now, we use radiation extensively in medicine, and it covers a lot of different fields of medicine. And X-ray, of course, is probably the most popular one that most people know about. But radioactivity is also used for many different types of scans. You guys can probably mention more than I can. It's also used in cancer treatment and a whole form of other different medical treatments. Now, we've been talking about atoms and isotopes and stuff like that, but now we come to the fact atoms join together to form molecules. And in order for them to join together, they have to form chemical bonds. So a chemical bond is something that holds atoms and molecules together. Now, there's different types of chemical bonds depending on how these molecules are held together. So we can have covalent bonding, which is the strongest, or forms a very strong bond. And this is formed when electrons between two atoms share, two atoms share electrons in their outer valence shells. So this is a covalent bond. It's very strong, and it's strongest between atoms that have similar electronegativities. And that's something that you'll get into in your chemistry course. We can also have ionic bonding. Now, this is a bond that's formed when two atoms exchange electrons. This allows for the creation of ions, for example. And ions exist in two forms, cation and anion. The cation has a positive charge, and the anion has a negative charge. Another type of bond we can have is hydrogen bonding, and this is formed by the attractive force between one electronegative atom and a hydrogen covalently bonded to another electronegative atom. We can see this in water molecule, for example, which is, to life, probably the most important molecule on the planet. So why are ions so important? Ions are groups of atoms that exist with either a positive or a negative charge. Generally, we get that by either adding or acquiring an extra electron or losing an extra or two electrons. Those are ions, and ions are super important when we're talking about biochemical reactions, and we'll talk more about those as we move through this unit. Electrolytes are compounds that are formed by ionic bonds, and they can release those ions when they're in solution. Electrolytes are very important in homeostasis, which is something we talked about in the last unit. Our body really needs to hold on to electrolytes and keep them in balance in order for a lot of our normal metabolic reactions to occur. So a lot of the energy that we expend in maintaining homeostasis is involved with maintaining electrolyte levels in various body fluids. They're also one of the most widely measured parameters in laboratory medicine. If you go to a doctor and you're not feeling well and he prescribes a urine test, for example, most of the things that we're going to measure in that urine test are going to be electrolyte levels. And based on the levels of those electrolytes, we can tell different things about different organ systems and their health. So a urine test is very, very valuable because it's cheap, it's really simple, it's non-invasive, doesn't hurt the patient, and we can get a lot of information about it just based on what's excreted in the urine. So just a couple of conditions here. Hyponatremia would be if a person has low sodium in their urine or in their blood. Hypochloremia, which would be low chloride. If a person is consistently measuring low potassium, then we would think about conditions or a condition or disease that could affect the heart, even cardiac arrest. So just by knowing and understanding the body's electrolytes compositions, we can get a good idea about the health of the patient. Now, mixtures, these are things that kind of come up in biochemistry, not as much as you'd see in chemistry, certainly, but it's a good idea to have some of these definitions. So a mixture is a blend of two or more substances, and we generally come up with three different mixtures. We have a solution, so that's a homogeneous mixture, so that means all the same, of one substance dissolved in another. So if you look at a solution, you wouldn't be able to tell that there's two compounds in there. Another one form of mixture is a suspension. Now this is a heterogeneous mixture, which means not all the same. That will mixture that will separate unless it's shaken up. And you can often find a precipitate, which is a solid forming within that mixture. The third one is a colloid, and the mixture is suspended, but the particles do not dissolve. They remain evenly distributed based on their small size. Now, solution is probably the most important one in terms of biological significance. So tend to focus on solution in biology. In order to understand solutions, we have 3 more terms there you should be aware of. We have the solvent, the solute, and an aqueous. A solvent is the substance in which something is dissolved. So the best way to understand solvent, solute is to think about a glass of water and you pour table salt in there. The water is the solvent, all right? That's the substance that something is going to dissolve into. The solute is going to be the salt. So we take our glass of water, we pour salt in there. The solvent is the water, the solute is the salt. If we mix it up, then we have a solution. We've got 2 different molecules and we can't tell them apart. They look completely the same. If the solvent in a mixture is water, then we say that solution, or that solution is aqueous. So aqueous generally means a water solvent. So hopefully those terms are clear, but I promise you when you do your chemistry courses, you'll get a lot more information on this. We're just presenting stuff to you now that'll give you a basic understanding for biology. Now, the importance of water cannot be stressed enough when we're talking about biology. It's the most abundant compound in the body. It is critical in all physiological processes. Dehydration, so that's a condition where the body has low water levels or low water composition, is deadly. If you're in a situation where you're lost and you need to find food or water, find water first. Okay, you can live a lot longer without food than you can without water. And water is fantastic because it exists in 3 different states on the planet. We have gas, which is water vapor, so steam. We have water as a liquid, which it exists at normal room temperature, and we have water as a solid. It can exist as ice. So it's a very special molecule. It participates in all body's chemical reactions, super important metabolically. And it's composed of 2 hydrogen atoms and an oxygen atom that are held together by covalent hydrogen bonds. Another couple of terms to throw at you: acids, bases, and salts. So when we're talking about acids and bases, we're talking about measuring hydrogen ions. So an acid is a substance that can easily donate a hydrogen ion to another substance. A base is a substance that can readily accept a hydrogen ion or donate a hydrogen ion. And then a salt is the compound that's formed between a reaction between an acid and a base. When we measure acids and bases, we measure them on the pH scale. pH scale is a measure of either acidity if the pH is low, or alkalinity if the pH is high. So the scale goes from 0, which is very acidic, to 14, which is very basic. And when we do measure pH, what we're measuring are the relative concentrations of hydrogen and hydrogen ions that are in a solution. Okay, so it's simple what a pH is. It's also a logarithmic calculation. So every time we move up a step on the pH scale, so we go from 2 to 3, that represents a 10 times increase in the number of hydrogen atoms. So it's a log scale, not a linear one. So that's also important to keep in mind. One of the things that our body does is it creates acids. A lot of our metabolic reactions will create acids, and this can have an effect on the pH of the blood, for example, which is something we need to keep very constant. So a buffer is a substance that prevents a sharp change in pH of a solution. Okay, now our body will produce buffers generally from bicarbonate or phosphate or a protein in nature, and we secrete them so that we can control pH. So the pH of the blood generally ranges from 7.35 to 7.45, so just slightly above neutral. If these pH ranges change really quickly, we can have very serious health risks, and we can even die from changes in blood pH. So buffers exist so that we can regulate changes in pH to maintain that range for our blood chemistry, so that we maintain homeostasis. Acid-base disturbances can lead to a bunch of different health conditions, and one of them is respiratory alkalosis.